The overall goal of this protocol is to demonstrate the villous growth of human intestinal cells under peristalsis-like motions and flow in a gut-on-a-chip microfluidic device, as well as to show how to co-culture these cells with living gut microbiome.
So this method can help the key questions in the field of the gastroenterology, clinical microbiology, and the pharmaceutical sciences to identify the disease mechanism and to validate the efficacy and toxicity of the new drug compounds.
The main advantage of this technique is that the researchers can establish the viable and functional host microbiome ecosystem of the human living intestine in vitro using a microphysiological system. The implications of this technique can extend towards diseases such as ulcerative colitis or Crohn's disease, since the stable co-culture of the host cells with the microbiome can emulate the complexities of these diseases. Though this method can provide an insight into the complex interactions between the gut microbiome and the immune system in the intestine, it can also be applied to other organ systems where there are host microbe interactions such as the skin, urogenital tract, or the oral cavity.
To begin, prepare the gut-on-a-chip microfluidic device as described in the accompanying text protocol. The device consists of a transparent, gas-permeable silicone polymer containing 2 parallel microchannels separated by a flexible porous PDMS membrane. On each side of the channels are vacuum chambers, which can be used to apply cyclic mechanical strain. Sterilize the device by using a 1 milliliter syringe to flow 70% ethanol through the channels.